So, uh, welcome to all of you again in today's session of this lecture, Electron Transport Chain. Earlier we have discussed a brief outline of the respiration and then two major complexes involved in the electron transport chain. So, today we will discuss in detail the other complexes involved in electron transport chain. So, as we have discussed that electron transport in the electron transport chain, different protein carriers are involved. We have discussed in our previous lecture complex 1 and 2, and then we have also discussed the differentiating point between these two complexes. So, further in this series of electron transfer and proton pumping, there is another complex third, also known as cytochrome C reductase or the cytochrome BC1 complex, which serves as a very crucial component of electron transport chain, and this complex is involved in the process of oxidative phosphorylation. Now, question arises where this complex is localized. Just I have shown in my earlier slides that this complex is also located in the inner mitochondrial membrane. You see this inner mitochondrial membrane, and then plays a very central role in the transfer of electrons from reduced coenzyme Q to cytochrome C, ultimately contributing the generation of ATP. Now, if we talk about the structure of this complex, that is cytochrome BC1 complex, this basically is a very large complex, and its structure was revealed by X-ray crystallography. Generally, much more was revealed in the between 1995 to 1998 through this technique. And you see that this complex is basically complex consists of 11 polypeptide subunits. With generally out of these 11 subunits, only one is generally encoded by mitochondrial genome. So, this is very interesting point to note to all of you. And this complex has a structure similar to cytochrome B6F complex present in the chloroplast. And this B6F complex serves as a part of photosynthetic electron transport chain. We have discussed in detail while discussing the photosynthesis. Now, what are the key structural component of cytochrome BC1 complex? It includes cytochrome B, cytochrome C1 and RSK iron sulfur protein. So, how this complex is involved in carrying out Q cycle and proton transport? You see that the mechanism of participation of ubiquinone in the electron transport process was first time proposed by Peter Mitchell, and Peter Mitchell termed it as a proton motive Q cycle. So, basically the Q cycle serves as a pivotal aspect of cellular respiration, which plays also a very crucial role in the electron transport chain within the mitochondria, where majority of the ATP production takes place. And this process is also very much integral to the energy generation in aerobic organisms, providing the energy necessary for various cellular activities. See, you see in the slide, at the heart of the Q cycle lies the enzyme complex cytochrome C oxidase, and that is found in the inner mitochondrial membrane. And the Q cycle basically involves the utilization of coenzyme Q. You see, coenzyme Q, also known as ubiquinone, and as an electron carrier, and then we will discuss how this initial electron transfer occurs. So, basically the process of electron transfer begins when the electrons derived from NADH or FADH2 are transferred to complex 1 or complex 2 of the electron transport chain. These complexes then pass the electrons to ubiquinone, converting it into ubiquinol or you can say QS2. Then how the electron enter into the Q pool? Basically ubiquinol, you see that ubiquinol carries the electrons to the Q pool, where it serves as a mobile carrier shuttling between complex 1 and complex 3 of the electron transport chain. Then the Q cycle begins. How this cycle begins? Basically at complex 3rd, this complex 3rd, the Q cycle starts at this here with the binding of you see that molecules of QS2 or ubiquinol, and one ubiquinol molecule donates basically 2 electrons to the cytochrome B subunit of complex 3rd, and then leading to its reduction to ubiquinol, or you can say Q here you see in this slide. Now how this complex 3rd is involved in proton pumping and different intermediate steps. So as you see that electrons are transferred through complex 3rd, basically protons are pumped across the inner mitochondrial membrane into the intermembrane space. You see here protons are pumped into the intermembrane space. So you see that this pumping of protons into the intermembrane space creates an electrochemical gradient. Then second round of electron transfer occurs, that the second ubiquinol, you see here ubiquinol molecule donates 2 electron to again another cytochrome B subunit, resulting in the reduction of another ubiquinol molecule into ubiquinol. However, this process, you see this process involves a transient intermediate state known as the semiquinone radical, this one, and then further it leads to regeneration of ubiquinol. The semiquinone basically radical formed in the previous steps interact with the another ubiquinone molecule to produce a stable ubiquinol molecule and leading to the culmination of Q cycle. Then electron transport continues. The ubiquinol produced re-enters into the Q pool, which is again ready to pick up more electrons and continue its role in the electron transport chain, whereas the cycle Q cycle further perpetuates. Basically the Q cycle not only facilitates the transfer of electrons, but also you see that contributes to the generation of proton gradient across the inner mitochondrial membrane, which is very much essential for synthesis of ATP through oxidative phosphorylation. Very interesting point I would like to share with all of you that Q cycle in mitochondrial electron transport chain is very similar to that occur in the photosynthetic electron transport chain in chloroplast. So this Q cycle is very much similar to mitochondria and chloroplast. Now complex 4, also known as cytochrome C oxidase, which is a very crucial component of oxidative phosphorylation process that occurs also again in the inner mitochondrial membrane of eukaryotic cells. It is the final complex in the electron transport chain responsible for catalyzing the transfer of electrons to molecular oxygen, ultimately leading to the formation of water. And first key features of complex 4 I will discuss one by one. Then first structure, how this complex is organized. So complex 4 is a large transmembrane protein complex composed of basically 13 subunits and contains 2 heme groups, heme A and heme A3, and 3 copper ions arranged as 2 copper centers. Copper center, basically copper A contains 2 copper ions linked by 2 bridging cysteine residues. The second copper center basically is QB, that is coordinated by 3 histidine residues, and basically these subunits work together to facilitate the transfer of electrons from cytochrome C to molecular oxygen, you see here. Then how this complex is helpful for transfer of electrons. Basically, electrons are delivered to complex 4 from cytochrome C, and then which carries them one at a time. The electron then pass through the several centers within this complex, including the copper ions and the heme groups in a generally series of redox reactions. Ultimately, you see that in the slide the electrons are transferred to the molecular oxygen, which serves as a final electron acceptor. Now, how this complex 4 is helpful in proton pumping activity? You see that proton pumping, generally the electrons are transferred to complex 4, and then this complex pump protons across the inner mitochondrial membrane from the matrix to the intermembrane space, and basically this process contributes to creation of proton gradient, or you can say proton motive force, which is generally used to drive the synthesis of ATP through ATP synthase complex. Now, how this compound is helpful in the reduction of oxygen? Generally, molecular oxygen serves as the terminal electron acceptor in the electron transport chain. You see that in complex 4, oxygen molecules are sequentially reduced to water through the addition of electrons and proton, and together heme A3 and copper B form the active center, and generally at which oxygen is reduced to water molecule. And this step is very much essential for the efficient disposal of electrons and the prevention of reactive oxygen species formation. Then how this complex is helpful in coupling of energy? Basically, what happens, the transfer of electrons to the complex 4 is generally coupled with pumping of protons across the inner mitochondrial membrane. This coupling of basically electron transfer and proton pumping creates an electrochemical gradient which stores potential energy that is used for harnessing ATP synthesis. Now, very crucial point I would like to share with you how this activity of this complex 4 is regulated. So, basically activity of complex 4 is very much tightly regulated to meet the energy demand of the cell. Various factors including the availability of substrate, pH and redox state of different electron carriers basically could influence the rate of electron transfer through the complex. So, basically this complex 4 plays a very crucial role in oxidative phosphorylation by catalyzing the final step in the electron transport chain, contributing to generation of ATP. So, basically it functions very much essential for maintaining cellular homeostasis and supporting various physiological processes. Now, you see that energy generated from moving 2 electrons downhill from NADS to oxygen creates 10 proton gradient across the inner mitochondrial membrane, generating a significant membrane potential. So, basically proton buildup causes a positive side of the membrane to face the intermembrane gap, while negative side faces the matrix. So, the proton gradient and voltage gradient work together to create proton motive force, which moves proton from cytosol to matrix. So, this is one aspect, because you see that the membrane is impermeable to protons and generally they pass via complex 4, also known as F0, F1 or ATP synthase, which is generally related to the production of ATP. So, the energy released during the movement of electrons is used to basically pump proton across the inner mitochondrial membrane, creating a proton gradient and voltage gradient. So, the combined proton motive force generated by these gradients is generally harnessed by ATP synthase to synthesize ATP, which serves as a key energy source, energy of the various cellular processes. Now, we have discussed the complexes involved in oxidative phosphorylation, which involves the transfer of electrons to oxygen and through complex 1, 3rd and 4th. So, basically this electron transfer you see is coupled with synthesis of ATP from ADP and inorganic phosphate by F0 F1 ATP synthase or complex 5. The efficiency of ATP synthesis basically depends on the nature of the electron donor. Like complex first is also known as ATP synthase or ATPase, is a very remarkable enzyme complex located in the inner mitochondrial membrane of eukaryotic cells. It plays a central role in the process of oxidative phosphorylation, which is responsible for majority of ATP production in the different kinds of aerobic organisms. So, at the heart of this complex 5 lies the ability to harness the proton gradient established by the electron transport chain to drive the synthesis of ATP from ADP and inorganic phosphate. Now, we will discuss in detail how the structure of this complex 5 is organized. So, complex, you see that in the slide that complex 5 is composed of two main components, F0, F1 and F0 complex. So, the F1 complex, this one which is localized towards the mitochondrial matrix is known as soluble catalytic domain, and another one is F0 complex, which is a membrane bound domain embedded in the proton translocating domain. So, out of these F0, F1, basically F1 complex, you see that in the slide, it protrudes into the mitochondrial matrix and responsible for synthesis of ATP. So, the F0 complex generally spans the inner mitochondrial membrane and forms a proton channel through which generally through this proton flows basically flows down their electrochemical gradient. And now question is, what is the main function of this F0 F1 ATP synthase? So, basically this is involved in the synthesis of ATP and from ADP and phosphate, and basically this synthesis of ATP occurs in this F1 complex which contains 3 catalytic sites. This F1 complex has 3 catalytic sites, namely alpha, beta and gamma subunits, and these subunits are responsible for synthesis of ATP. Then this is also helpful in translocation of protons. So, generally as you see that protons flow through the F0 complex from intermediate space to the mitochondrial matrix. You see that protons flow through this F0 complex to mitochondrial matrix. The F0 rotor subunit, this F0 rotor subunits basically rotates, and this rotation is basically coupled with conformational change also in the F1 complex, and which enables it to synthesize ATP from ADP and phosphate. Now, how this F0 F1 ATP synthase helps in the synthesis of ATP? So, basically movement of how this movement of proton occurs. Generally protons from the intermembrane space enter into the F0 complex through a channel protein formed by subunit C, while it simultaneously drive the rotation of the rotor subunit. You see, the rotor subunits rotate, and the rotation of this rotor subunit induce a conformational change in the catalytic site of F1 complex, facilitating the binding of ADP and inorganic phosphate to synthesize ATP. Then how ATP is released from this complex? So once ATP is synthesized, it is released from the catalytic site into mitochondrial matrix. You see in the slide with yellow color, where it is could be utilized as energy source for carrying out various cellular processes. Now, how the functioning of this complex is regulated? Generally, complex 5 is regulated to match the cellular energy demands. For example, if there is any increase in the concentration of ADP, which is to match the synthesis of ATP by complex 5 to meet the increased demand for ATP. Opposite to it, if generally high concentration of ATP inhibits the activity of complex 5, and once its level is high, basically it prevents unnecessary synthesis of ATP and thereby conserving the cellular energy. So basically this complex 4 is a very remarkable machine, you can say, that serves as a primary ATP synthase in most of the eukaryotic cells. Its ability to harness the proton gradient generated by electron transport chain to drive ATP synthesis is you can say very crucial and plays central role in cellular energy metabolism. So basically understanding the structure, function and regulation of complex 5 is essential for understanding the mechanism of oxidative phosphorylation and its further significance in the various physiological processes. Now, in this I am going to discuss the role of electrochemical potential gradient in facilitating the movement of various molecules involved in citric acid cycle and ATP synthesis within the mitochondria. Now, what is the role of electrochemical proton gradient? Question arises. Basically the electrochemical proton gradient is involved in the movement of organic acids of the citric acid cycle and substrate or products for ATP synthesis in and out of the mitochondria. Now, how ATP is synthesized and transported? Basically ATP is synthesized in the mitochondrial matrix, but you see that most of it is used outside the mitochondria. So basically ADP and ATP transporter is responsible for the active exchange of ADP and ATP across the inner mitochondrial membrane. Then how movement of ATP in and out of mitochondria takes place? Basically this is the proton driven transport. The movement of ATP out of the mitochondria in exchange for ADP is driven by the electrical potential gradient. Positive is outside and generally generated by proton pumping activity. Then uptake of phosphate. Active phosphate transport involves a protein that is used in the chemical potential component delta pH of the proton motive force to drive electroneutral exchange of inorganic phosphate in for OH out. Then how pyruvate is taken up? Uptake of basically pyruvate is driven by electroneutral exchange with hydroxyl ion leading to the continued uptake from the cytosol. Now, very interesting fact, what is the energetic cost? The energetic cost of taking of one phosphate and one ADP into matrix and exporting one ATP involves the movement of one proton from the inner mitochondrial membrane into the matrix, and basically electroneutral exchange of hydroxyl and phosphate consumes chemical potential component, that is delta pH, but not the electrical transmembrane potential component delta E. So the total cost is 3 protons used by ATP synthase plus 1 proton for exchange across the membrane with a total of 4 protons. Then how organic acids are transported? Then the inner mitochondrial membrane basically contains transporters for dicarboxylic acid, basically malate or succinate, then exchange for phosphate and for tricarboxylic acid like citrate, aconitase or isocitrate which is also exchanged for dicarboxylic acid. So, you see that the electrochemical proton gradient basically drives the movement of molecules crucial for citric acid cycle and ATP synthesis within the mitochondria, uh, highlighting the importance of various transporter in this crucial process. Now, complete oxidation of a sucrose molecule, you see in this slide, results in the net formation of how many molecules of ATP through substrate level phosphorylation. You see this 4 from glycolysis and 4 from tricarboxylic acid cycle, total 8 molecule. 4 molecules of you see that NADPH are produced in the cytosol, you see this during this process. Additionally, 16 molecules of NADH and 4 molecules of FADH2 are generated in the mitochondrial matrix via succinate dehydrogenase. And now we will discuss what is the theoretical generation of ATP. Basically, theoretical ADP and oxygen values are basically used to estimate the 52 molecules of ATP will be generated per molecule of sucrose through oxidative phosphorylation. Now, how much total ATP is synthesized? So, the complete aerobic oxidation of sucrose in this you see in the slide, including substrate level phosphorylation, results in the total of 60 ATP synthesized per sucrose molecule. Now, we will compare the energy efficiency. This basically amount represents about how much 52% of standard free energy available from the complete oxidation of sucrose, with the rest being lost as in the form of heat. So, basically this represents a significant improvement over the fermentative metabolism, where only 4% of the energy is available in sucrose is converted into ATP. But you see during the oxidative phosphorylation, electrons are transferred to oxygen through complex first, third and fourth, and this process is basically linked to the production of ATP from ADP and phosphate through the F0 F1 ATP synthase, or you can say complex 5, and the quantity of ATP generated varies basically based on the type of electron donor involved. Now, electron donors and ADP oxygen ratios is very crucial. Electrons donated to complex 1, for example, from malate oxidation, results in ADP oxygen ratio of 2.4 to 2.7 with an average of 2.5. Electrons donated to complex 2 from succinate and external NADH dehydrogenase generally yield ADP and oxygen ratio in the range of 1.62 to 1.8 and average is 1.5 here you see. So, direct donation to cytochrome C oxidase complex, that is complex 4, via artificial electron carriers gives ADP oxygen ratio of 0.8 to 9, 0.8 to 0.9. Then proton pumping and ATP synthesis is also very crucial. Experimental ADP and oxygen ratios align well with values based on the number of protons and pumped by complex 1st, 2nd and 4th, and the cost of 4 proton is associated with producing 1 ATP. So, in summary, you see that the energetic efficiency of sucrose oxidation through aerobic respiration highlighted the ATP production, NADH and FADH2 generation, and theoretical yield and the conservation of free energy. Also, it emphasizes the efficiency of aerobic respiration compared to fermentative metabolism in utilizing the energy stored in sucrose. Now, these are the various inhibitors involved in electron transport chain. I will discuss one by one. One is rotenone. Rotenone basically inhibits complex 1. So, you must note at this point which specific electron inhibitor is inhibiting which complex. So, like rotenone inhibits basically complex 1 of the electron transport chain. Generally it binds to ubiquinone binding site, preventing the transfer of electrons from NADH to ubiquinone. Then another one is antimycin C inhibits complex 3rd. So you should note it very specifically, cytochrome complex 3rd or cytochrome BC1 complex of the electron transport chain. Basically this inhibitor binds to Q1 site within complex 3rd, blocking the transfer of electrons from ubiquinol to cytochrome C. Then third one is cyanide, which inhibits the complex 4 of the electron transport chain, and it basically binds to heme cofactor within cytochrome C oxidase, blocking the final step of electron transfer to oxygen, which prevents the formation of water molecule. Then carbon monoxide is another inhibitor. Like cyanide, carbon monoxide binds to heme group in cytochrome C and block the transfer of electrons in the electron transport chain. Another one is azide here. Azide is a reversible inhibitor of complex 4. It competes with oxygen for the binding to heme group in cytochrome C oxidase and blocking the electron transfer to oxygen. Then amobarbital, also known as amital, inhibits complex 1. Then oligomycin also inhibit complex 5, that is ATP synthase of the electron transport zone. So you should note here at this is very crucial that which inhibitor is inhibiting which particular steps. So these inhibitors serve as basically valuable tools in biochemical research for studying the mechanism of electron transport and ATP synthesis. And some of these inhibitors have clinical significance, are used in various purposes for the treatment of various metabolic diseases or poisoning cases. Now another aspect is what is the respiratory quotient, abbreviated as RQ. So respiratory quotient is a ratio that indicates the relative amount of carbon dioxide produced and oxygen consumed during the process of cellular respiration. So basically it is a measure of type of fuel being utilized by the cells for the production of energy, and the respiratory quotient is calculated as the ratio of the volume of the carbon dioxide produced to the volume of oxygen consumed. So this respiratory quotient could provide valuable information about the substrate being metabolized for energy, and different types of compounds like carbohydrate, fats and proteins have different respiratory quotient. And the respiratory quotient can provide, as I mentioned, very important information for the kind of substrate being utilized for generating metabolic energy. And these different types of compounds like carbohydrate, fats and protein have distinct respiratory quotient, as I mentioned. And like carbohydrate, for example, you see carbohydrate have RQ close to 1. So basically carbohydrates are primarily oxidized in the presence of oxygen, producing equal amount of carbon dioxide and oxygen. That's why its RQ is 1. For fats, you see that RQ for fats is lower than 0.7, as fats require more oxygen for their metabolism, and the combustion of fatty acids generates more carbon dioxide relative to oxygen consumed. That's why its RQ is less than 1. Now what is the RQ for proteins? RQ for proteins varies, but it is generally between 0.7 to 1. The exact value of RQ for protein depends on the specific amino acid and composition of proteins that is being metabolized. This is the general formula for calculating the RQ. As you see that the basic equation for aerobic respiration involves the breakdown of glucose molecule into carbon dioxide and water, and energy is produced in this reaction. So in this reaction 6 molecule of carbon dioxide are produced and 6 molecule of O2 is generally consumed, and so RQ for this reaction is basically 1. So I must thank you to all of you for going into details of these aspects, and hope you will enjoy the respiratory quotient, electron transport chain. I discussed in detail the different complexes involved in electron transport chain, and their specific features of these electron complexes I have discussed in detail, and how these complexes they differ from each other, which crucial aspect they play. So all these aspects I have discussed in detail. Hope you might have enjoyed this lecture. Thank you very much.